Hi, my name is Manish Butte, and I'm an immunologist here at UCLA and a professor in the Department of Pediatrics and Microbiology, Immunology, and Molecular Genetics. My lab focuses on three large areas of research. First, I'm not only a professor, but I'm also a doctor here, and I take care of children and adults with rare genetic immune diseases. We're very interested in these patients who come to us with infections and autoimmunity because they have some defects in the genes that control their immune system. The more that we can study why these kids get sick, the more we actually learn about how the immune system works. And the lab also has been focusing for many years to understand how mechanical forces affect the immune system. Everything from swelling of tissues to the trafficking of cells as they move throughout our body is influenced by the forces of tissues and other cells nearby. And what we've come to learn is that this is a mechanism by which the body can regulate the immune system during infections and afterwards. Another area that we've been working on is harnessing the immune system to fight cancers. And we've been developing a series of biomaterials and nanomedicines in the broader term to try to amplify the immune response to be able to fight cancers. And we've been developing other areas as well, including in studying the metabolism of immune cells in cancer, and even a new project that's developing a new type of vaccine for infections. Overall, the theme for all of these projects is a type of cell called a T cell. My lab is very focused on T cells, trying to understand fundamentally how they work and how we can harness them to understand and treat diseases. So in many of our patients, many of the patients who come to us have rare immune diseases. As I mentioned, these are experiments of nature, patients who are born with errors in their immune system. And my lab has been working very hard to identify new diseases. And this is an example of a little girl that I've been following for many years who we identified with a new type of genetic disease. And we published her story a number of years ago. Her clinical phenotype was that she had a lot of swelling in her lymph nodes all the time. And other doctors thought that she had some kind of cancer, but she didn't have a cancer. She had a genetic disease. And because we were able to figure out the cause of it, we actually came up with a new treatment for her. This little boy came to UCLA with a severe infection, and I'm going to show you more about him in the next slides. We were able to come up with a whole new treatment by repurposing his immune system with drugs that we took from other fields and pushing his immune system to be able to fight infections better. And finally, we've been working with mice, and we're trying to create these mice to actually have human immune systems in them. Not healthy immune systems. We're actually recreating the immune systems from children that we see so that we can actually do experiments and understand how immune diseases work by studying the animals that recreate the human diseases. In this way, we really can accelerate diagnosis and treatment of patients without having to go to them to draw blood and do biopsies and other invasive procedures. So this is that same boy, and we published his case about a year ago now, 6 months ago to a year, where we were able to treat his severe fungal infection called coccidioidomycosis by taking medicines that were invented for other diseases, dupilumab, which was invented for allergies, and interferon, which was originally invented for cancer and for a neutrophil disease. And we were able to treat him with these medicines in a way that allowed his own immune system to wake up and fight the infection, an otherwise fatal infection. This is another example of how we repurpose drugs. This is a little girl who was born with an inborn error of her mast cells, where she produced this severe, blistering, debilitating disease as an infant. We were able to talk to Novartis and get them to share a new drug with us and treat this girl and monitor her so that we really eliminated her symptoms, and she's able to grow up to be a normal toddler. Another part of my lab, as I mentioned, is developing biomaterials to fight cancer. One of our projects is involved with breast cancer. And as you may know, breast cancer affects about 1 in 8 women in the United States at this point. In particular, we've developed a biomaterial that looks here like a little sponge. It's biocompatible and biodegrades naturally into the body. But when a surgeon plants it next to the tumor, before it degrades, it activates the immune system locally and brings T cells in to fight the cancer. And we've been able to show in mouse models of cancer that this therapy has a lot of promise. We're now working with the Johnson Comprehensive Cancer Center funding to try to move this therapy into breast cancer models in mice and eventually to humans. And finally, my lab has been working on a very fundamental basis of understanding how T cells turn on and turn off. As I mentioned in the introduction slide, we're very interested in how mechanical forces at the molecular, cellular, and tissue scales actually drive and control the immune system. For example, we were able to develop a whole new type of microscope called an atomic force microscope and measure the forces generated by individual molecules on T cells as they push and pull in their environment. We're actually able to prove that T cells generate forces and receive forces, and those forces are necessary for the T cells to become activated. This is a whole new paradigm of receptor signaling to try to understand not just that the receptor needs its ligand to bind and activate, but the ligand has to come with force attached to it. We were able to actually use this discovery in a subsequent paper published just in the last year for translational purposes. We actually have made an apparatus where we can drive T cells with a mechanical shaking force and turn and supercharge them. This kind of capability will be very important as we and other doctors start using T cells to fight diseases like cancer. If we can supercharge the T cells before they're placed into the patient to fight the cancer, we think that the treatments will be more effective. These projects have been funded by the National Institutes of Health. Thank you for your attention.